Good afternoon, everyone. My name is Wenjing Wang, and today I would like to talk about our work on the genetic regulation of the human skeletal muscle in response to weight loss. And we can moving from the book level transcriptomics and QTL mapping to single nucleus resolution. And finally, we can also discuss about the spatial transcriptomics. So here, then the central question behind this work is how do genetic variants and lifestyle intervention interact to shape the skeletal muscle biology? And can this help explain why individuals respond differently to weight loss? Here, the talk has three parts. First, I will introduce our published book RNA-seq and the QTL mapping study. Second, I will show our ongoing single-nucleus RNA sequencing atlas. And finally, I will discuss how spatial transcriptomics can help us to place this molecular change back into the tissue context. I will start with the first part, our book RNA-seq and QTL mapping work. This part focuses on the skeletal muscle transcriptomics regulation before and after lifestyle intervention. This study was published in Cell Genomics in 2025. Here, we used paired skeletal muscle biopsies from 54 participants who underwent a 16-week lifestyle intervention, and we asked how gene expression, splicing, and genetic regulatory effects change after weight loss. And to begin with, I would like to show you the broader background, where obesity and type 2 diabetes are shaped by both genetics factors and environmental or lifestyle factors. Where then we know that the genetics actually this is have some information will have impact on the phenotype. This is well known, and the lifestyle we can control by ourselves. And while the environmental actually are usually largely non-modifiable. And on the genetics side, there's been really important foundational work in this space. Like Suzuki and colleagues explored the genetic drivers of type 2 diabetes heterogeneity, and Sparkling and colleagues identified type 2 diabetes associated loci in over 400,000 East Asians, which is particularly relevant given the most genetic studies and have a historically focused in the European populations. And Scott and colleagues, as well as the GTX consortium, have mapped how the genetic variants regulate the gene expression. And you know that the large-scale GWAS studies have identified the mailing loci associated with obesity, insulin resistance, and type 2 diabetes. These studies have greatly improved our understanding of diseases risk. At the same time, molecular QTLs, like including this GTX and related work, have shown that mailing diseases association variants may act by regulating gene expression or splicing in relevant tissues. So we already know that genetics matters. And we also know that gene regulation is one important mechanism linking the genetic variants to complex diseases. And on the intervention side, Ligard and the colleagues showed that exercise combined with diet-induced weight loss improved beta cell function, while the Mills and colleagues found that weight loss combined with exercise enhanced metabolic function in people with pre-diabetes. However, most genetic studies and most lifestyle intervention studies are still considered separately. Genetic studies usually ask which variants increase the disease's risk, and lifestyle studies usually ask, does the intervention improve the metabolic health? But in the real life, these two factors are not independent. A person's genetic background may influence how they respond to the lifestyle change. So this brings us to the key gap. Some effects cannot be explained by genetics alone, and some effects cannot be explained by the lifestyle alone. And the phenotypical levels, we often see very different responses to the similar intervention. For example, after similar calorie restriction or exercise program, some individuals lose a lot of weight while others lose much less. Similarly, some individuals improve insulin sensitivity dramatically while others show a weaker response. And the molecular level, this leads to and important questions. How do diet, exercise, and weight loss influence the genetic regulation? In other words, can lifestyle intervention modify the effect of the genetic variants on gene expression and splicing? This is the gene by lifestyle interaction questions that motivated our study. And here, this is a nice reference study from Cell Genomics that published in 2024, showing that genetic regulatory effects can be modified by diet. They used bamboos because this kind of the study is very hard. It's very hard to do in humans. You need long-term dietary intervention, repeated phenotyping, and access to the living tissues. The important concept here is the genetic effects are context dependent. A variant may have a one regular effect under one dietary condition, but a different effect after metabolic stress or lifestyle change. For human obesity and diabetes, we ultimately need the human data. This is where our cohort becomes valuable. We have paired human skeletal muscle lab biopsies before and after our lifestyle-induced weight loss intervention, which give us a rare opportunity to study the lifestyle-responsive genetic regulation directly in human muscle. The cohort we use here is the Singapore Adult Metabolism Study 2, or we call it the SAMS2. This was a very challenging human intervention study. The study started with 265 recruits after screening, eligibility assessment, intervention completion, biopsy collection, and sequencing quality control. The final cohort included 54 participants. And you can see where we collected the muscle biopsy samples before and after the intervention here in phase 1 and phase 3. And this may sound like we only got like a very small number, only 54 in the final cohort compared to with large population scale genetic studies. But for a human skeletal muscle intervention study with paired biopsies, this is actually a very valuable cohort. It took around 3 years to complete the full study, and the key strength is the pair design, and each participant had the skeletal muscle biopsies before and after the same 16-week lifestyle intervention. This allows each individual to serve as their own control. So SAMS2 actually provides like an ideal design to study the gene by lifestyle interaction in human skeletal muscle. And this slide shows the main biology motivation. All participants underwent the same 16-week lifestyle intervention, but their outcomes were very different. Some participants showed strong improvement in insulin sensitivity, where then you can see it on the right panel. And while others had much weaker responses. This is important because like skeletal muscle is responsible for a large proportion of insulin-stimulated glucose disposal. So change in the skeletal muscle biology may directly related to the change in whole body metabolic health. And here, our hypothesis was that genetic variants may modulate the transcriptomic responses in skeletal muscle. And this may help explain why different individuals respond differently to the same intervention. And here we first look at the differential gene expression before and after intervention, where then we identified the 505 differential expressed genes, including 364 upregulated genes and 141 downregulated genes after intervention. The enriched pathways were very consistent with the metabolic improvement. We saw upregulation of mitochondrial biogenesis and oxidative phosphorylation, insulin signaling, and the GLUT4 mediated glucose uptake, lipid metabolism, and well as the myofiber remodeling and this kind of the sarcomere organization. So overall, you can see like lifestyle intervention appears to activate a coordinated metabolic program in skeletal muscle. And this gives us the first layer of the information, which genes respond to the intervention. and the bulk tissue level. And next, we perform the molecular QTL mapping. We mapped the eQTL and sQTL separately before and after intervention. For eQTLs, we identified over thousands of the E genes, both pre-intervention and the post-intervention. And for sQTL, we identified the hundreds of the significant S intron both pre-intervention and after intervention. The key point here is that some QTLs were shared between the two states, while others appear to be pre-specific or post-specific. This state-dependent QTLs suggest that lifestyle intervention can modify genetic regulation of gene expression and the splicing. So the same genetic variant may have the different regulatory effects depending on the metabolic or lifestyle state of the individual. We also ask whether some of these QTLs are specific to the Asian populations, because our cohort actually is all complete from the Asian populations. And here, this is a very important thing, because then most of the existing QTL resources are still largely based on the European ancestry individuals. And here we compared our QTL results summary statistics with the GTEx skeletal muscle dataset, where then we found that around 4.2% of the eQTLs and 7.3% of the sQTLs showed the Asian specificity. And although the percentage is not very large, and these signals are important because they may be missed in European-focused references datasets. And this highlights the value of the building ancestry-diverse molecular QTL resources, and especially for the tissues like skeletal muscle that are highly relevant to the metabolic diseases. Here's one interesting example, which is the gene actin-3. Actin-3 is a well-known skeletal muscle gene involved in the muscle function and the fiber performance. In our study, we found that this is a shared eQTL before and after intervention involving the actin-3 that was associated with the individual weight loss response after intervention, where you can see the individual with the TT genotype actually have a more like lower weight loss change compared to the other two genotypes, the CC and CT for this gene, for this variant. And this is a nice example because it links three layers together: the genetic variation, skeletal muscle gene regulation, and the clinical response to lifestyle intervention. And of course, this does not mean actin-3 alone explains the entire response. Weight loss and insulin sensitivity are complex traits, but it provides a concrete example of how genetic regulatory effects may contribute to the inter-individual variation in intervention outcomes. And beyond the shared QTLs, we were particularly interested in the QTLs and the change between the pre and the post-intervention states. These are the QTLs that may reflect the gene by lifestyle interaction. For example, a variant may modulate a gene strongly before the intervention, but weakly after the intervention, or vice versa. This effect suggests that the regulatory landscape of the skeletal muscle is dynamic. It is not fixed. It can shift with the metabolic state and the lifestyle intervention. This is one of the main messages from the bulk QTL study. Like lifestyle intervention does not change gene expression levels. It may also change how genetic variants regulate the transcriptome. And we then integrated our QTL results with metabolic GWAS signals based on the Asian populations using the localization analysis. The goal was to identify the diseases associated loci where then the GWAS signal and the skeletal muscle QTL signal may share the same causal variants. And through this analysis, in total, we identified 16 putative metabolic risk genes. This suggests that some obesity or diabetes associated loci may act through the gene regulation in skeletal muscle. This is important because GWAS loci often point to broad genomics regions, but they do not directly tell us which gene or which tissue is involved. And by combining the GWAS with skeletal muscle QTLs, we can move closer to identifying the candidate efforts, effect genes, and the regulatory mechanisms. And this bulk RNA-seq and QTL mapping work has now been published in Cell Genomics. But this study also made one limitation very clear. Bulk RNA-seq gives us an average signal across the whole tissue. It does not tell us which cell types are driving the observed change. That brings us to the next part of this project. And this slide summarizes the key findings from the part 1, where the book level data. And like we identified 500 differential expressed genes after lifestyle intervention. We identified more than like thousands of the E genes and hundreds of the S introns, including like hundreds of the lifestyle responsive and Asian-specific regulatory effects. And we also highlighted some interesting examples. For example, like the actin-3 weird and genetic regulation may be linked to the individual response to the intervention. And finally, through the GWAS colonization, we also identified the 16 putative metabolic risk genes. However, the major limitation is that the bulk RNA-seq reports a population average, where we see a DEG in bulk muscle. We do not know then whether it comes from like which cell type, the type 1 fibers, or like immune cells, in stromal cells, or other cell populations. So the next question is, which cell type are responsible for these bulk transcriptomic signals? To answer this, we need the single-nucleus resolution. Now I would like to move to the second part, our single-nucleus RNA-seq atlas. This is an ongoing project where we're providing the skeletal muscle across lean, obese, and weight loss conditions. The goal is to resolve the cell type-specific transcriptomics change and eventually cell type-specific genetic regulation in human skeletal muscle. And for the single-nucleus RNA sequencing projects, we combined two complementary cohorts. The first is the cross-sectional cohort, which includes the lean and the obese individuals from the 3 Asian ancestry, Chinese, Malay, and Indian. This cohort contains 205 skeletal muscle biopsies. The second one is what we show actually in the Cell Genomics paper, which is the same SAMS2 cohort, where then includes 54 obese individual samples before and after a 16-week lifestyle intervention. So in total, then this gives us 108 biopsies. And together we have a 313 skeletal muscle biopsies from the 269 individuals. And this design gives us two types of the biological contrast. The cross-sectional cohort allows us to compare lean and obese muscle across individuals and the ancestries. And the longitudinal cohort allows us to study the within individual change after weight loss. And we also can ask both what differs between lean and obese muscle and what changed after intervention. And we already sequenced a total of the greater than 2 million nuclei. And where after the strict QC, and then we left about 1 million high quality nuclei for the follow-up analysis. So, like conceptually, this project has these 3 layers. And the first layer is what I talk about in the Cell Genomics paper, which tell us then which genes and variants are involved. And the second layer is a single-nucleus RNA sequencing, which tell us which cell types are responsible. And the third layer is spatial transcriptomics, which tell us where these cells and the molecular chains are located inside the tissue. This multi-resolution strategy is important because each layer answers different questions. And spatial data here then also can give us some tissue architecture, and the single nuclei data gives us some cell type resolution. Um, so here we know that the skeletal muscle is a very focused metabolic tissue. It accounts for a large proportion of the insulin-stimulated glucose disposal and the primary site of the GLUT for mediated glucose uptake. And then compared to the available data sets and also the literature review we have done for now, and we also noticed that there is still a major gap, except for our cell genomics paper. And we also notice lifestyle responsive regulation has mostly been studied at the bulk level, but not actually in the single cell or spatial level data. And the cell type specific and the spatial genetic regulation in response to the lifestyle intervention remains largely unexplored. And this is especially true in Asian populations. So here then our goal is to build a single nucleus and spatially resolved references the human skeletal muscle across obesity and weight loss states. And here, after quality control, we obtained around 1 million nuclei and identified the 19 major cell types here. This includes the major myofibrous populations, such as the type 1, type 2A, and type 2X fibers. We also identified stromal cells, including like FAPs, tenocytes, and smooth muscles. We identified some vascular and the immune populations, and expected the dominant cell types are the myofiber nuclei, but we are also very interested in some non-myofiber population, especially the FAPs, because it may play important roles in adipogenesis, fibrosis, regeneration, or metabolic remodeling. One here of the first thing that we examined was the fiber type composition. In the obesity comparison, we observed that the proportion of the type 2X fibers increased with the BMI and decreased with the improved insulin sensitivity. And in the weight loss cohort, after intervention, we observed a decrease in the type 2X fibers and increased in the type 2A fibers. This suggests a shift from the more glycolytic fiber state towards a more oxidative fiber state. And biologically, this is very consistent with the metabolic improvement, where then we also use our available clinic traits and to do the correlation with the cell type proportion. We also identify the very consistent pattern, where then the type 2X fibers are more glycolytic, while type 2A fibers have a more oxidative capacity. So this 2X to 2A shift may represent fiber remodeling after intervention. And of course, with the single nucleus data, we need to be cautious about the technical effects in cell proportions estimation, but the direction of the change is biologically meaningful and consistent with the improved skeletal muscle metabolic function. And next, we asked which cell types drive the bulk transcriptomic signals. One clear finding is that mainly bulk signals are driven by the myofibers and by the cell type, which actually account for a large part in total of the nuclei. So this actually is expected because muscle fibers and the FAPs dominate the tissue. For example, this gene NR4A3, which actually is a well-known exercise and inactivity responsive gene, is consistently downregulated in type 1, type 2A, and type 2X. That's all three muscle fibers in obesity. This suggests that some obesity-associated bulk signals are fiber intrinsic, but not all the bulk signals come from the myofibers. For example, another gene, the GRB14, which is related to the insulin resistance, show an obesity-associated signal enriched in the FAPs and the adipocytes. Even more interesting, some of the top bulk upregulated genes after intervention are highly enriched in the myofibers and the FAPs. This means that if we only look at the bulk RNA-seq, we might think that these genes represent the general muscle response, but single nucleus data shows that part of the intervention responses may actually reflect the stroma or FAP remodeling. And we also observed regulation of the WNT signaling in adipocytes and FAPs. Since then, the WNT signaling can suppress the adipogenesis. This may suggest that after weight loss intervention, adipocytes and the FAPs shift away from the adipogenic program. So the key message here is that the single nucleus RNA sequencing helps us reinterpret bulk findings and the cell type resolution. And currently, we are working on the cell type specific QTL calling and the context specific QTL calling, and to see if then the bulk level and the single nuclear RNA sequencing level data can share part of the QTL results. And we also will have more power to find it, and because we will call the QTLs by cell type population. And just based on these very preliminary results, and we can see actually there are a lot of the interesting examples then to explore in the following work. And now I would like to move to the final part, the spatial transcriptomics, and also discuss some potential directions that we are working on now. Then a single nucleus RNA-seq give us cell types and the cell states, but it does not tell us where those cells are located in the tissue. For skeletal muscle, this spatial information is very important because tissue architecture is closely linked to the function. And the skeletal muscle actually is a very classical multi-nuclei tissues. So our next step is to place these cell type specific signals back into the intact skeletal muscle tissues. A single nucleus RNA sequencing have one major limitation, where then the tissue dissociation destroys the spatial information. And after dissociation, we can only identify the cell types and the transcriptomic states, but we lose their physical location. This means that we cannot directly observe whether FAPs or the muscle fibers close to the specific cell type, whether the immune cells accumulate in certain tissue regions, or whether obesity-associated changes occur near vascular and like adipocyte infiltration or fibrotic areas. And here, the spatial transcriptomics can address this limitation. It allows us to measure the gene expression in situ within the tissue context. In this project, we are building the platforms using the Wenjing MERFISH and also with the 10x with an HD. These platforms will allow us to validate the cell identities, localization, condition, association, and test whether predict cell-to-cell interactions are physically plausibly. And here then we choose this tool, the Wenjing MERFISH, which actually is the imaging-based in situ spatial transcriptomics, while then the 10x with an HD sequencing-based spatial transcriptomics also can provide us some additional information, especially if we would like to know more information about like splicing or isoform level thing. And here, there were three main questions, and we hope to answer with the spatial transcriptomics. The first question is whether the novel or the rare populations identified in single nucleus RNA-seq correspond to a real, spatially distinct populations in intact muscle fibers. And for example, if we identify the rare clusters in single nuclear RNA sequencing, spatial data can help determine whether it represents a real biological structure or a technical artifact. The second question is where, like the obesity associated or the weight loss associated transcriptional changes occur. And are these changes fiber intrinsic, or are they located near the blood vessels, or are they enriched in like a spatial cell type? Are they associated with like adipocyte enriched or the fiber enriched regions? So this is very interesting because then we have very comprehensive and uh, and complementary cohorts where then we can compare in these two cohorts and also validate in each other. And the third question is about the cell-to-cell interactions. Like if a ligand receptor analysis predicts the interaction between the FAPs and the myofibers, spatial data can tell us whether these cells are actually close enough to interact. This is particularly important for interpreting like the spatial cell type biology and understanding how stroma remodeling may influence the muscle metabolisms. So, to bring everything together, our long-term goal is to build a comprehensive regulatory references for the Asian human skeletal muscle. The first layer is the bulk RNA-seq and the QTL mapping. This tells us which genes respond to lifestyle intervention and which variants regulate the expression and splicing. And the second layer is the single-nucleus RNA-seq. This tells us which cell type are responsible for these signals. For example, we see the fiber type remodeling from type 2X to type 2A. We see the FAP-associated interventional signals. And the third layer is the spatial transcriptomics. This will tell us where these cell types and the molecular programs are located in the intact tissue and whether predicted interactions are spatially meaningful. So together, these three layers allow us to move from like which genes change, which cell type drive this change, and where does this happen in the tissue. So ultimately, we hope this work will provide the first longitudinal ancestry-diverse, spatially resolved regulatory map of the Asian human skeletal muscle and help us to understand why individuals differ in their metabolic response to the lifestyle intervention. So with that, finally, I'd like to thank everyone in our lab who contribute to this work.